Hi everyone, welcome back. Today's video is about the polypeptide structures. Before we get to the video, please make sure to subscribe to our channel as this helps us out a lot. Okay, let's get to the video. Proteins are complex macromolecules made up of amino acids. Their structure can be described at different levels. The different levels include the primary, secondary, tertiary, and quaternary structures. Let's consider each in more detail. Primary structure. The primary structure of proteins refer to the linear sequence of amino acids in polypeptide chains. It is the most basic level of protein structure. The primary structure is essentially a list of amino acids linked together in a specific order. This order is determined by the genetic material of an organism. This may be DNA or RNA. The genetic code determines the sequence of amino acids in a protein. Therefore, any changes in the DNA sequence can result in a different amino acid being incorporated into the protein. This affects its structure and function. Refer to transcription and translation to learn more about the process of how DNA or RNA genetic material is used to make a protein. The primary sequence that is created is unique to that specific protein. There are 20 different amino acids commonly found in proteins. These amino acids are the building blocks that make the primary structure of polypeptide chains. They are linked together by peptide bonds. Peptide bonds are covalent bonds. They link the amino group of one amino acid to the carboxyl group of the adjacent amino acid. The sequence of amino acids is written from the N-terminus to the C-terminus. This sequence is critical because it dictates how the protein will fold into higher order structures and ultimately determine function. Now let's consider the secondary structure. Secondary structures of proteins refer to local, repetitive folding patterns that occur within a segment of the polypeptide chain. These structures are stabilized primarily by hydrogen bonds between the amino hydrogen of one peptide bond and the carboxyl oxygen of another. The two main types of secondary structures are alpha helices and beta sheets. Alpha helices are right-handed coiled structures, while beta-pleated sheets are extended sheet-like arrangements. Let's look at each. Alpha helices are right-handed coils or helical structures formed by a single extended polypeptide chain. This helical structure is stabilized by hydrogen bonds between the carbonyl oxygen of one amino acid and the amino hydrogen of an amino acid located 4 amino acid residues down the chain. The helix is compact and stabilized. Alpha helices are common in regions of proteins that require stability, such as in the core of a globular protein. Beta sheets. A beta sheet is a more extended structure where the polypeptide chain folds back and forth. This forms a sheet-like arrangement. The adjacent strand can run parallel or antiparallel to each other. Beta sheets are stabilized by hydrogen bonds between amino acids in different strands. The alternating pattern of hydrogen-bonded amino acids give rise to the characteristic pleated appearance of beta sheets. So sometimes beta sheets are also called beta-pleated sheets. Beta sheets are often found at the core of globular proteins as well, and in certain structural motifs. There are also beta turns and loops. Beta turns and loops are regions in the polypeptide chain that change direction. They often connect different secondary structures together. Beta turns have a tight loop structure and are stabilized by hydrogen bonds as well. And loops are more extended regions that connect secondary structures. Secondary structures contribute to the overall three-dimensional folding of proteins, which means that they contribute to tertiary structures. Therefore, they play a crucial role in determining the protein's function. The specific arrangement of alpha helices, beta sheets, and turns contribute to the stability, flexibility, and functionality of proteins. Now let's consider tertiary structures. Tertiary structures refer to the overall three-dimensional arrangement of the protein's polypeptide chain. Tertiary structures give a protein its unique and specific 3D shape, and it is crucial for the protein's function. As you may have noticed by now, I've repeated that structure equals function, and that is key to note in biology as well as biochemistry, especially in the context of proteins. Structure equals function. If something disrupts structure, that disrupts function. Tertiary structures is the result of interactions between the side chain of amino acids. That means tertiary structures result due to interactions between the R group. Interactions between R groups can be one of four types: hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bonds. Hydrophobic interactions are between nonpolar amino acid side chains. Nonpolar amino acid side chains tend to cluster together to minimize exposure to water. Hydrogen bonds are formed between polar side chains, and on the other hand, ionic bonds occur between charged amino acid side chains. And finally, disulfide bonds form covalent bonds between sulfur atoms of two cysteine residues. It forms a disulfide bridge. Tertiary structures can also be classified as globular structures and fibrous structures. Globular tertiary structures refer to proteins that are compact and are rounded in shape. This type of structure is common in enzymes and many soluble proteins. Fibrous tertiary structures refer to proteins with elongated or fibrous-looking shapes. These types of proteins often have a structural role. For example, collagen is a protein that has a fibrous tertiary structure. When it comes to tertiary structures, prosthetic groups are something that needs to be discussed. Some proteins require non-amino acid components called prosthetic groups to achieve their complete tertiary structure and therefore perform its function. Examples of prosthetic groups include metal ions or coenzymes. Tertiary structures also determines the location and conformation of the functional site on a protein. These sites are often involved in binding to other molecules, catalyzing biochemical reactions, or other specific types of functions. When we talk about proteins, denaturation of proteins is something that comes up often. When we refer to denaturation of a protein, tertiary structures are what gets disrupted. Tertiary structures can be disrupted by extreme conditions such as high temperature, changes in pH, or the presence of certain chemicals. This disruption is what is known as denaturation, and it leads to the loss of a protein's biological activity. And finally, there is the quaternary structure of proteins. Quaternary structures refer to the arrangement of multiple polypeptide chains into functional higher-order structures. These polypeptide chains are often referred to as protein subunits. Proteins with quaternary structures consist of two or more polypeptide chains where each has its own tertiary structure. So essentially, the quaternary structure describes how these subunits interact with each other to form a complex. There can be different types of interactions between these subunits. They include hydrophobic interactions, hydrogen interactions, ionic bonds, and disulfide bonds. Hydrophobic interactions occur between nonpolar regions of the subunits and may cluster together to minimize exposure to water. Hydrogen bonds are formed between polar amino acids in different subunits. Ionic bonds occur between charged amino acids from different subunits. And disulfide bonds are covalent bonds that form between cysteine residues of different subunits. Quaternary structures of proteins can be one of two types: heteromeric complexes and homomeric complexes. In heteromeric complexes, the subunits are different from each other. Each subunit within the heteromeric complex may have different amino acid sequences, tertiary structures, and functions. Hemoglobin is a classic example of a heteromeric complex. It consists of 4 subunits, each with a different amino acid sequence. In the case of hemoglobin, the subunits are labeled as alpha and beta globins. This forms a heterotetramer. It has 2 alpha subunits and 2 beta subunits. In homomeric complexes, the subunits are identical or very similar to each other. Therefore, the subunits of homomeric complexes are often referred to as protomers. An example of a homomeric complex is ferritin. Ferritin is a complex that stores iron. Ferritin forms a hollow sphere consisting of 24 identical subunits. Each subunit contributes to the overall structure of the complex, and all subunits are the same. And that concludes this video. I hope this video was helpful for you. If it was, please make sure to like and subscribe to the channel. See you in the next video. Bye!